Yeah, good morning. Thank you for having me here at the BioEM Talks, and I will talk about high pressure freezing. Try not to get too much in the weeds. Kind of give a kind of first half will be about high pressure freezing, how it works, and the second bit will be a kind of a case study. So yeah, this background. I've been doing EM for quite some time. My first introduction to EM was in 1995 when I was an undergraduate at Penn State and with Larry Eckroth. And then in grad school, I worked with Robbie Robertson at ASU. And then kind of my EM career solidified my time at University of Colorado Boulder, working with Andrew Stalin, Dick McIntosh, Tom Giddings, and the rest of the 3D EM lab folks. And then when I started here at UChicago in 2005, I spent some time with Kent McDonald out in Berkeley, Debbie Sherman at Purdue, and Howard Berg at the Donald Danforth Plant Center, kind of learning how to run a core and organize things. And then I've been here, as I said, at University of Chicago since 2005, and I'm the director of the Advanced Electron Microscopy facility. Our facility has grown over the last couple of years, and so we've just kind of created a new EM suite. We have a Titan Krios. We have a volume scope for large volume imaging, Aqualos 2 for cryofit milling, and just added a Glacio. So we have this new facility. We work with internal users and external users. So if anything I talk about is interesting, reach out. We can see if we can help with your research. Our goal here is to, and my driving goal is to take biological problems, analyze them in a Google Earth manner by obtaining a large tissue view and then zooming in to visualize intercellular compartments and ultimately attaining the structures of specific macromolecular components at angstrom resolutions. Before I get started with my talk, I do want to take a moment and kind of in remembrance of Andrew Stalin. This is the first talk I think I've given since he passed away last year. And so Andrew was, he started his career as a grad student with Hans Mohr, studying who pioneered freeze fracture techniques. We've heard some of that in the talks previously. He came to America in the 70s, kind of started his career at University of Colorado Boulder, really pioneering the best ultimate fidelity for cellular structures. And then later in his career, when I was a postdoc in his lab, he was deep into tomographic reconstructions. Two projects that we worked on. One was the thylakoid membrane system in chloroplasts, and the other one was plant cytokinesis and cell wall and membrane dynamics. So this talk's dedicated to Andrew. So to get started, I think the driving principle for all electron microscopists is to make a phenomenon visible as immersely to enlarge our power of understanding it. And this kind of, gets to the question of how do you study cells? I'm a cell biologist, and so this is there. And electron microscopy is kind of the technique in the 30s. Ruska developed a technique to use electron microscopy. And then starting from the 30s into the 40s, 50s, 60s, all the way through probably until the 1990s, EM was the kind of the go-to technique to study cells. Probably in the 90s is when light microscopy techniques took over. And ultimately, people thought EM had, as a technique, was finished and really had no place in cell biology except for maybe a pretty picture. And a lot of this was kind of due to chemical fixation and the artifacts that it presents. So when you take a cell, you add glutaraldehyde and other fixatives, you change the morphology of the cell. And then when you compare that to living or light microscopy techniques, you see something different. So this commentary came out in 2013 asking this kind of a rhetorical question, is EM dead? And then in 2017, we had the revolution. The Nobel Prize in cryo-EM was awarded. And then everyone got super excited. And, you get back to this point of how do biochemists study cells? They make the soup out of it and study the individual components. But that still doesn't get us to the context of what's inside the cell and kind of how do we get back to studying things, proteins, molecular machines within the context of the cell and the tissue. And so you have to run through how do we actually prepare the biological specimens for the electron microscopy. And you have to run through these steps. You have to fix it somehow. You have to then remove kind of the aqueous water to get it ready to go into the vacuum of the electron microscope. Typically, you have to embed it in something. If you're doing plastic, you could go directly to the EM now with cryo. Then you have to section it, maybe post-staining it, and then eventually look at it. And so before we go in, it was mentioned in the last talk that, ChemFix is still widely used and maybe, the favorite technique. And I think a lot of people shy away from HPF because it sounds difficult, or it could be a little challenging at times. But I kind of want to go through why not chemical fixation, because that's the question I get asked when I present HPF as an alternative. And if you go through this process where you're looking at how to go from the cell, the fresh cells to something we study in electron microscope. If we look at the example here, by the time you run through your chemical fixation process, you no longer have a pristine sample. You've compromised that sample in many ways through the fixation and then ultimately through the dehydration processes. And just to illustrate that, Kellenberger has a nice work done on this from in the 80s. But this shows that as you apply chemicals, what you get is that you start to bind up your globular proteins, or globulous and fibrous protein structures start to come together, or your fibrous protein structures will kind of bind into these kind of unnatural states, which we want to avoid if we're trying to study the cell in its live state. And so let's tackle fixation. The best way we can think of to preserve it, as Dick McIntosh said, in a live state, is to use cryo techniques. And so here's this kind of guide. There's a number of cryofixation techniques. We have plunge spray, propane jet. These were, Andrew Stalin was, and folks at ETH Zurich, they really pioneered a lot of these techniques. The cold metal slammer, we heard about that in a previous talk by John Heuser and Tom Rees, were kind of pioneering that. And then high pressure freezing, once again, that was kind of came out of the ETH Zurich kind of group in Europe. And then Andrew kind of brought these techniques here to America. And once you get it frozen, there's a number of paths you can take. We heard today about freeze fracture. I'm going to talk a little bit about getting into the TM. We now have cryo ET. So this is kind of the cryofixation is the way to go. How do you choose between all these cryofixation options? We heard a little bit in the previous one. Essentially, it's about cell thickness or sample thickness. Plunge freezing and the jet freezing methods, even the slammer freezing methods, they have a thickness limit of about 40 to 50 microns. And that's because you can't remove heat fast enough from the sample to actually prevent ice crystal formation. And so that's where we go to high pressure freezing and using that as a technique. And there's several HPFs out there. Just to get this out the way, this is the one we have at UChicago, HPM010. It's no longer manufactured. Commercially you can get the Volvin instrument, and actually historically Volvin was involved in the engineering of the 010. So the Volvin instrument is very similar and works very similarly to the classic HPM010. And then you have the Leica Ice, which is a pretty new model, pretty slick, and has some really nice features. So just to recap, the differences about conventional fixing and rapid freezing. One, it's speed. Conventional fixation is extremely slow, seconds to hours to actually fix the cell or tissue, whereas rapid freezing is very fast. The high pressure freezing, you fix the cell in about 3 milliseconds. Specificity with ChemFix, it's a very selective cross-linking. Every chemical has its favorite target. So, osmium is unsaturated double bonds, glutaraldehyde is, protein. So you have this kind of selective kind of cross-linking. So you don't fix everything at once, whereas in rapid freezing, you fix all molecules instantaneously. Applications, I think with ChemFix, there could be a case made for low, medium resolution kind of studies, qualitative studies, whereas rapid freezing, you could do qualitative studies, but you can also now do high resolution kind of studies and maintaining cellular fidelity. The size is the only advantage of conventional fixation. You could do really large samples, millimeters and up, whole organs, you can actually chem fix is dump it into a vat of aldehydes and fix it nice and slow. Whereas with rapid freezing, as we heard, you are limited to smaller samples and maybe the upper range of 600 microns or so. How does high pressure freezing work? We heard a little bit about that in the last talk, so I won't belabor it too much, but essentially you raise the pressure to about 2100 bar, which slows the expansion of ice and reduces the formation of ice crystals. And that it also lowers the freezing point of the cell by about 20 degrees Celsius. So you can freeze very rapidly this thick tissue, thicker sample, and not generate damaging ice crystals. Theoretically, you could get to about 1.5 millimeters, but generally to play it safe, or what I've seen in my hands, 300 microns to 600 microns is kind of the sweet spot in terms of thickness for high pressure freezing. And just an example, here's a filamentous cyanobacterium, Anabina. We have the vegetative cell here that does photosynthesis. We have a heterocyst here that fixes nitrogen. This was kind of the standard image that were being generated all the way up until 2010. We still saw images like this. Went through this with the group here, Bob Hazelcorn's group at U Chicago. And, this is the heterocyst after doing high pressure freezing, working out the protocols, where now we have the vegetative cell. We can see the thylakoid membranes very clearly. We can see the heterocyst. We've maintained the outer cell wall layers, the polysaccharide layer, the glycolipid layer. So you're going to see just time and time again that when you apply high pressure freezing or cryofixation techniques, you're going to have a cellular fidelity that's much higher and more reliable than ChemFix. So the entire process, as C3PO said, he should be quite well protected if he survives the freezing process, that is. That's the big trick here about cryopreservation, is actually getting a sample frozen without generating damaging ice crystals. And so in the sample, we can actually see ice damage. And so as you form ice nucleation inside the cell during freezing, you can imagine that what you're doing is actually bringing water and dehydrating the sample in a way where you're kind of bringing all of the cellular components into some compact kind of form. And so as we go through the process where you move the vitreous or the ice out and replace it with resin, when we get to the TEM, what we see is this kind of spiderwebby look here. And so here's an example of a well-frozen mouse cerebellum. And this here is a not well-frozen mouse cerebellum. This one is pretty extreme, and you can see that this looks really bad. But sometimes the damage is a little more subtle, so limited to maybe the nucleus. And so the nuclei have lower solute concentrations, and so they're much more susceptible to ice damage. And so here the cellular components look relatively well frozen, but the nucleus has obviously suffered from a lot of ice damage. So as you analyze your images, you do have to be conscious and thinking about, is the sample have ice damage or not, as you go through. And then just kind of, I'm kind of really just giving an overview. If you're tackling high pressure freezing or you're stuck, please reach out. I've done quite a bit, and so happy to help. But just some general rules that you can follow to really make your high pressure freezing run successful. One is to start with healthy samples. The cells and tissues must be in the optimal physiological condition you want to study. Because you freeze so quickly, you really want the material to be at the place and conditions you want to study. So if you bring half-dead cells, you're going to be studying some half-dead biological state. So that's kind of important. Work quickly. You have to go from your living tissue to the frozen cells. Ideally, in less than a minute should be your goal. Sometimes, though, it's not possible if you have dissections, things like that, or you're adding drugs to the sample, or you're trying to do some study. You might delay a little bit, but really you want to minimize that time and go as fast as possible between the live cell and frozen cell. Biggie is don't let your cells, tissues dry out. If you're working with a cell culture material, you got to use a humidity chamber maybe, or put something around, or use Petri dishes filled with water and wetted paper. to help keep your sample hydrated and humid. Avoid mechanical damage, sloppy cutting, overfilling the specimen cups. We saw in last talk, if the specimen cups were shown, if you overfill that, or if it's not kind of neatly done, then that will cause ice damage in poorly frozen samples. Use the smallest volume of sample you can. Even though I said you can go up to 600 microns worth of sample, if you can get away with 100 microns of sample, that's ultimately going to give you a higher level of success than trying to push and get the largest volume possible. And then the last one is to use a filler to remove air spaces. So if your sample goes into the planchette, the sample holder, and it has air spaces around it, remember, air is an insulator and will prevent the sample from cooling at the most optimal rate and giving you a nice vitreous sample. And so we have to think about filling that space with something. We can't just use normal buffers because the solute concentration is too low. So generally we use things like 0.15 molar sucrose, hexadecene, dextran, phycol, even yeast paste to fill up the spaces around. And there's many of these fillers. Once again, depending on your sample, you would kind of cater that and kind of design as to fit your sample really well so that it freezes best. So that's kind of the fixation run. It's how do we get the sample from live to frozen. So now we have the sample. It's vitreously fixed, hopefully, with vitreous ice. And so the next question is, what do we do with it? If you're going directly to the TEM in a cryo-ET type application, you would just take the sample and go to a cryo-fib mill and do cryotomography. But if you're going to do plastic work and kind of do ultrastructure or plastic tomography, you have to dehydrate the sample still. And this process is what we call freeze substitution. So we keep the sample at minus 80 degrees C, and then slowly replace the vitreous water ice with a solvent. And this process can be done in a machine, like here's the Leica AFS machine that we use, or it can be done in a dry ice box. So when I was at Boulder, we didn't have fancy machines. We just got a box of dry ice, and that was close to minus 80 C, and we did our free substitution protocol on that. And even now, Rick Webb and Kent McDonald have their fast freeze sub process, which is done in a dry ice box also. And I think some company makes something, EMS or Ted Pella makes a free substitution box. The solvent you use, acetone is usually the favorite. But there are protocols that include methanol or ethanol. You have to be a little careful with the methanol and ethanol because they can extract lipids. And so you could get a sample which you have a lot of lipid extraction if you add too much of these methanol or ethanol. We didn't add our fixatives or metals in the free sub material. So in the acetone, we would usually add our glutaraldehyde, uranyl acetate, or even osmium tetroxide. And so as you freeze sub, the idea is that as you're removing the vitreous ice with the solvent, that you're introducing now your fixative, glutaraldehyde or osmium, so that once the temperature raises a little bit to minus 50 C, once the chemistry starts in earnest, then you'll get these binding events and stabilize your sample. And then you will find protocols where there's additives. So, some folks add a little bit of water to the sample. They think that helps with their staining. There's, protocols that add tannic acid as a mordant and helps with binding of osmium. I've played around with these things and they can help, but they also can leave some artifact behind. So you could get kind of some precipitates and things if you don't wash out the tannic acid completely, that's unbound. So you do have to be careful there. So there are a lot of parameters that you can play around with to get the sample to work, but you have to go in and kind of think about every step you can modify the protocol a little bit to be tailored to your sample that you're trying to image. And so just an example of this is when I started working on a chloroplast from Arabidopsis, I followed the normal protocol that was established in Andrew's lab. And unfortunately, the sample came out where the membranes were not stained very well. And actually, it's a pretty poor sample, even though it was very well frozen in the vitreous ice. There was no ice damage, but we did not get a lot of staining with that. But playing around with the protocol, as I said, kind of playing with the conditions, actually was kind of removing the osmium at different time points and doing different washings seemed to really help the sample. And so here now you can make out, and these are plastic tomographic images of a spinach chloroplast. There were Arabidopsis also, but we can now see the membranes. We see this very nice kind of the helical structure that the chloroplast thylakoids are famous for. And ultimately, if you take some of these preps and you compare them now to more recently cryo-ET, cryofib milled samples, you can actually make out even the molecular components. So in this plastic tomogram high pressure frozen sample, you can make out the oxygen evolving complex as you can in this cryo-fib mill sample. So, this goes to show that, to get high resolution information, you don't always have to go to cryo-ET. High pressure frozen free substituted plastic tomogram can get you really close to the answers that you need and not go through some of the struggles to get a milled sample ready for the cryos to do cryo-ET. So just kind of something to think about. I think people have forgotten the power or don't know the power of high pressure freezing in plastic tomography. So after that, we go from dehydration. Our sample now, it's dehydrated, it has a solvent in there, metals. Now we go to the embedding step. There are a number of choices here to also be made. Your sample can be warmed to room temperature, so through slowly from -50 up to room temperature, and you can use a classic resin, EPON, Spurs, whatever your favorite may be. Or what we do a lot of is that we hold our sample at -50 and we embed with a lauryl resin, generally HM20, but we have used K4M also for different applications. But HM20 is the best. If you're going to do CLEM, correlative light electron microscopy experiments, you would definitely go into a lauryl resin and hold it at that temperature to do those types of experiments. And then the rest of the process, thin sectioning, staining, TM is exactly like you would follow for a ChemFix protocol. So really, the work is done up at the top, introducing cryofixation methods, and then free substitution, and then kind of choices of embedding. And so just kind of want to go through quickly. I've done many samples over my career, my 18 years at UChicago, all sorts of things from plant, bacteria, mammalian samples. Each one is a little unique and have their kind of thing. So this is a leaf cross section. So I've showed chloroplasts many times, but we can do entire leaf, put leaf pieces in and get these really nice cross sections across. Here are some airway smooth muscle. These were held at physiological length. And so just really nice kind of preparations. You can see these very long, smooth muscle cells. We can go in and see the actin filaments in these cells and nicely preserved. We can take tissue samples. So this is liver tissue. We can see red blood cells. We can see what we believe is an NKT cell kind of cruising through the tissue. And so you can see here in these two examples of different places in the tissue, we can see this nice nucleus has been preserved really well. You can actually see probably nuclear pores right here. The condensed chromatin there. So really nice preservation across different organisms, tissues. And even we can look at this was an entire colony of bacteria. So it was picked off the plate. And we used kind of a non-traditional carrier, a 6 millimeter carrier for our HPF. And so we had a little bigger. So usually the carrier is about 3 millimeters. But we've kind of modified my machine, and now the Leica Ice, I believe, and the Volvin, you can use 6 millimeter carriers. But this was a study looking at sporulation across the bacterial colony. And this is the prep. And here you can see at higher, you can actually make out the lipid bilayer, the little cell wall, and then the sporulation event starting to happen within the bacterium. And then, lastly, I know a lot of folks grow cells on sapphire, which is really nice, but they can be a pain to work with. And so, a lot of researchers were already growing cells on transwell. So they have these little Corning transwell inserts into their culture dishes. and they would grow their cells on top of these, or their primary cells. And what we found and played around with is that we could cut out these inserts, use the 6 millimeter carrier, and then just freeze that directly. And then we're not worried about damaging our diamond knife with the sapphire or removing the sapphire. We just kind of freeze this entire disc. And so here's like two examples. One are these glycoxyl layer. This was a study looking at the glycoxyl layer. I'm not showing it in here of endothelial cells, but here's the layering that we got off of that. And this was a mammalian primary cell culture grown on trans wells. So they kind of laid them down, and here's this nice kind of this actin field. I think this group was looking at tight junctions, so we were kind of looking at the cells that are kind of together grown on this layer. And so just kind of want to go through once again, summarize that earlier part, that there are a lot of steps in this that make it seem tricky, and there is some playing around. But we're at a point where probably if you have a tissue or something you're trying to study, if you reach out, there's a good chance that it's already been high pressure frozen at least once or twice in literature and or just kind of in general knowledge of how to approach it. So if you're stuck with your HPF, reach out, and always happy to help and give advice. And just kind of go back over sample prep. You want to study, work quickly. Know your sample. Is it prone to mechanical damage or drying out? Then you have to take steps to prevent that. Use the smallest sample possible. Use the correct filler and sample holder. Free substitution choices. You have a plethora of metals you can use. You can add mordants. You can add acetone, methanol, water. Generally speaking, we use uranoacetate, 0.1% uranoacetate and acetone. And I think most of the images I showed, unless I've noted it on a slide, they were done with uranoacetate and acetone only in the free substitution. And then the temperature, you can free sub at -90, free sub at -80. There are differences. We have seen some tissues respond better with a -90 free sub versus a -80. And so that's kind of the, something to play around with. But generally, most things are -80. And then, you use a free substitution machine or a box of dry ice. They both can work. And I've had success with both, and users have had success with both. And then the resins, once again, you can bring your sample to room temperature in a controlled way, and you can use spurs, EPON, or you can do low temperature embedding if you're doing CLEM applications. Mostly, as I said, we use uranoacetate, acetone, and then we stay at -50 and go into HM20. That is kind of our standard go-to protocol. For other things, we are, and what I'll talk about in the next half, is modifications to that to do other applications besides plastic tomography, plastic immunocytochemistry, or CLEM techniques. And so kind of with that part there, I always like this quote, your machinery is too much. I think as we switch gears and we start thinking about the larger picture, especially as we move from the resolution revolution into the context of the cell, I think this quote really fits the cell and the tissue and how it's put together. The machinery is too much. And it particularly, I like this quote because this applies so well to biology in the cell, but it's taken out of context and kind of using this to make a point. This is actually a line from a poem by Allen Ginsberg titled America, and it's about the machinery of America, not the machinery of the cell. And so I like that because if we take things and we study them out of context of the cell, of the tissue, of the organ, of the organism, we didn't miss kind of how these things function, and then we miss how we can actually leverage our knowledge to kind of disease treatment and other things like that. So kind of want to pull us back, and how can we combine high pressure freezing, get this really high cellular fidelity with kind of now a large volume EM tomography, so EM, large volume. EM just is one of the techniques of the year this year. I think Naomi had mentioned that her group, they do a lot of large volume work. So I kind of wanted to touch on this and how a project came where we couldn't use traditional chemfix. We had to use HPF. And so kind of just how we put everything into context. And so this project here, it's studying, it's an embryology kind of project. This researcher, they were looking at the kind of molecular cellular mechanisms, how tissues are patterned during the embryogenesis process, how developmental programs orchestrated across different physical cellular environments. You start out, you have to form the limb, the brains, the face, a plan. And the idea is to, if we learn things, can we get this developmental foundation of how this process works, kind of to use that for prevention strategies and even therapies for tissue regeneration and repair. So really kind of an exciting project. Here was just kind of just some info. A lot of this group's work was with, this is Tim Sanders' work, who is now at the UC San Francisco. They're studying this the chick embryo. You have this limb formation, and early in embryogenesis, with their light microscopy, they would see these specialized filopodia called cynemes. And these structures can be seen actually in this video, kind of wiggling their way through space, finding connections and making connections and connection events with other cells in this kind of three-dimensional embryonic space, which then will lay the plan for what these cells will become, how to become it, and body organization. And so this is pretty exciting. And, the question when I met with Tim was, how do we bridge the gap between cellular ultrastructure in the light microscopy and molecular resolution of the EM? And so up to this point, really, the only thing that had been done was kind of this kind of low resolution SEM work here, where you can kind of maybe make out cynemes and cells, but not very good, especially when you compare that to the light microscopy, things you can do with 2-photon microscopy and the live imaging. So how do you get there? How can we actually get to see the molecular components? And so, people have tried. This isn't the first time someone said, oh, we should use EM on this. But unfortunately, here's a kind of that live image again, no fix. What happens is when you add fixative, glutaraldehyde, even just a low kind of percentage, you begin to see the disruption of these cynemes, and kind of they break apart and they disappear. And what this did was lead to confusion in the field, because people said, well, maybe they're not real because we can't see them with EM and fixation. And it really then was like, no, but now we got these great live light techniques and we can see them, so they must be real. And so when Tim came to me, I said, well, let's do, and we looked at EM of ChemFix just to see that and prove to ourselves that you do indeed get these little fragments, and the cells don't look very happy. But I said, well, let's go to high pressure freezing, free sub. We worked out some conditions that worked. So we got lovely cell preservation, but also we could see the cynemes kind of still attached to the cells, still kind of reaching out. And here you can see a bunch of them in this field of view here, the slice, kind of still maintain that presence. We actually see extracellular matrix kind of components starting to form, which is really cool because, at this developmental age, that would just start to happen. So it was really neat to see these things in a cell and preserve this cellular fidelity using a high pressure freezing and freeze substitution. And then the next step was to move that to a serial block phase tomogram. And so essentially this is a process where you have an ultramicrotome in the SEM, and you take a slice of the block and you image the face, and you do that consecutively to build up a larger volume. And so there was a protocol already existing by Ellisman group, looking at, this was all in brain tissue, looking at using volume techniques or using high pressure freezing, and then combined with volume techniques. And so we had to modify that. Once again, I'll stress that as you do high pressure freezing, freeze cell, you generally, if it works, great to pull a protocol and let's go with it. But ultimately, you do have to have a little bit of a wiggle room and playing around And so we actually modified the percentage of osmium. We added some UA, a little more glute in our protocol. And then we used, we reduced the metal on the back end on the protocol. And then we used carbon black conductive resin because the lipid content isn't as high as in brain tissue. So that really helped with some of our imaging. And this volume, it's about 650 nanometer sections at 20 nanometer resolution. We did this at low vac. It took about 4 days. And what you can see is go back to what you can see here is you can see these like cynemes, these cells you can follow them and track them through the material. So really neat. And this was the first time at this level of resolution where you can actually see, the cellular context of these cynemes kind of going through the material and even forming attachments or connecting or these cynemes kind of sliding against each other or laying against each other as you go through the volume. So this is a really exciting project. This was, newish data. And so this is just a higher mag. And just to reinforce this idea is by combining the preservation power of high pressure freezing, freeze substitution with the large volume imaging technique, we finally have the ability to study kind of fragile cellular structures in the context of tissues at EM resolutions. And I think also this project goes to the larger scheme where a lot of folks now want to jump directly to cryo-electron tomography, yet they really have no understanding of what the cellular context is. So it's kind of like, let's just go collect a bunch of tomograms and see what we see. And somewhat now really educating folks that we should take a step back and really go and get these kind of very informative large volume views to really then pinpoint where we need to go or what we really want to study and see as we move forward with cryo electron tomography. So hopefully, high pressure freezing will kind of really be that plug-in between kind of the single particle SPA work and then cryotomography, molecular tomography kind of work. With that, just kind of want to give just some acknowledgments. Here, especially the team, it's just 5 of us. So Ime Chen, Josh Fisher, James Fuller, and Tara Lavoie. They kind of really are solid and great collaborators and to work with and run the facility. The signing project, as I mentioned, was Tim Sanders and Josh. Before he came over to the core, he was a work in Tim's lab, and so now he's part of our core. So. So that's the end of the talk. I could have gotten the weeds on any one of these topics, but wanted to give this just kind of quick overview because I love it. This is the 1,000,000-volt microscope that used to be at Boulder. I did get to use it, so I just like showing it, and it's fun. But I kind of leave with this last thought: could thou and I, with fate, conspire to grasp this sorry scheme of things entire? And with that, I'm happy to take any questions.